Scientists at UCSD essentially reversed Parkinson's disease in mice by turning astrocytes, which are the support cells of the brain, into neurons. Hi, Sri here, and welcome back to The Cutting Edge. You know, even the simplest hand wave can seem like a miracle when you think about the intricate sequence one's brain follows to make it happen. There's the planning of the wave in the frontal cortex, which gets shot to the basal ganglia deep in the brain, which acts like a control tower, giving the go-ahead back to the motor cortex, which then shoots signals down the spine and out to the arms' muscles, allowing them to move. And the fact that all of those messages, little packages of neurotransmitters like dopamine, are being shuttled from one neuron to another in fractions of a second is pretty miraculous. But if you're thinking that this entire sequence sounds like it can go really wrong, you would be absolutely right. In fact, in Parkinson's disease, the control tower of the operation, the basal ganglia, goes down. Specifically, neurons in this area called the substantia nigra, which normally produce dopamine, start dying off. And without enough dopamine, the little packages can't be sent between neurons, which leads to tremors, slow movement, rigid muscles, and unstable posture, characteristic of Parkinson's. But in Nature this week, scientists reported being able to fix this in mice by figuring out how to replace the dying neurons using nearby support cells called astrocytes. To do this, these authors focused on a protein found in astrocytes called PTB, a protein which they found is actually necessary for astrocytes to stay astrocytes. In fact, without it, astrocytes turn into neurons. So to replace dying neurons, these authors injected a small piece of RNA into the surrounding astrocytes, which basically took the RNA that would have become PTB off the market. These astrocytes turned into neurons, started making dopamine, and bada bing, bada boom. The mice started being able to move in a controlled way again. Now, whether or not this is an approach that could be used in humans is still super up in the air, but this is a huge first step, which granted can sometimes seem a little bit small in the grand scheme of the immense complexity of the brain, but hey, as George Pugh put it, if the human brain were so simple that we could understand it, we would be so simple that we couldn't. And that's the cutting edge.